Great. Well, good morning, everyone, or almost early afternoon. Thanks for coming in person. I know everyone's emerging from COVID slowly, so it's great to see those of you that made it. My name is Clint Elge. I'm a bioinformatics computational biology student here at the University of Idaho, and I am presenting. Very glad we finally got our guest speaker, Krisandra Allen, out here. We've been trying to get her out here for at least 6 months, but because of COVID, it's been very difficult. Krisandra is actually a UIdaho graduate, and after that, she went to the Virginia Commonwealth University, where she got a master's in public health. She has worked for the Virginia and the Washington State Departments of Health. Initially, she worked with foodborne pathogens and waterborne pathogens, and she was part of the transition to next generation sequencing with that. And then more recently, she got roped into COVID work with the Washington Department of Health. And just in the last month, she's made the jump to the private industry. She's now working for Biobot Analytics, which is a company that, among other things, does wastewater surveillance for SARS-CoV-2 and is actually doing samplings here in Moscow. So with that, I will turn the time over to Krisandra.
All right, well, thank you very much. I am really excited to be back in Moscow. Had a great time in my years here and really excited to see some of my favorite professors here today. So it's exciting to be on this side of this go around. So let's see if my clicker wants to work. All right, so I'm going to start off by telling a story of what got me really interested in how we can use molecular data for public health investigations. So I started out at a local health department in Virginia in the middle of nowhere, literally in a place called Farmville. And I got a call from the state central office that said, hey, you have a person in your jurisdiction with salmonella, and they are a match to this national cluster that we think is associated with tuna. Can you call them back and go through this special questionnaire to see if they match this recall that's going on? And I had initially, when I had talked to the patient, they didn't report any tuna or sushi or any of that. So I wasn't too hopeful, but I called back and started going through the questions one by one, and that jogged their memory. And they said, oh yeah, my friend had bought sushi. I never eat sushi, but they had bought some and I tried a bite. And went further through the questions, and sure enough, they had eaten the very specific type of spicy tuna roll, which was associated with this outbreak. So we went out and did the environmental health investigation and were able to trace it back and link it. And that was the moment when I figured out that we could tell by the DNA of the bacteria in somebody's poop what they had eaten and use that for public health purposes. That was my aha moment of, this is what I want to do. This is so cool. So I'm going to tell a lot of stories about foodborne outbreaks along the way, because that's what I've done for most of my career, and then transition into some of the COVID work that I did in Washington State. So I started in public health in 2012, and at that point, we were generally not doing any sequencing. It was all using PFGE. So you would use restriction enzymes to cut up the genome and then run it out on a gel and get these, what we would call DNA fingerprints, and then line those DNA fingerprints up next to each other to see which isolates were close enough to each other that they might have some epidemiologic link. So that's been used in public health since the 90s. The PulseNet system is what it's called, that coordinates the foodborne and enteric bacteria, was first started in 1996. So that was the technology in use when I first started my career. And then, as Clint mentioned, we have transitioned over time into using next generation sequencing for these foodborne and enteric pathogens, and even additional pathogens of public health interest. And one of the great things about using sequencing versus PFGE is that you can have a lot more confidence in the clusters. So sometimes we would get PFGE clusters and we could just never find anything in common, or we would have an outbreak at a known restaurant and we would get different PFGE patterns, even though we knew that they all ate the same implicated food. So there was a lot of noise in the cluster detection by PFGE. So moving to sequencing really gave a lot more confidence in the clusters, so we could focus our efforts better. So one of the things transitioning from PFGE to sequencing is, we call this our investigation triad. We have the lab, the epidemiologists, and the environmental health groups. And what happened when we switched to sequencing was those groups stayed the same, but in addition, we added the bioinformatics piece, which is not something that had really been involved in public health investigations up to this point. And so I want to talk about, this is an example of a Listeria outbreak. Let me see if I can shrink, there we go. Get rid of that. All right, so this is one of the outbreaks that really catalyzed moving to sequencing for Listeria investigations. So Listeria was the organism that kind of went first for a few reasons. One is because it has a much higher case fatality rate than Salmonella, E. coli, some of the other enterics. And it also has much smaller numbers. So it was a much easier task than trying to convert thousands of Salmonella every year. So this is an example of a cantaloupe outbreak in 2011. You can see there were 33 deaths involved in this outbreak. And one thing I want to point out is that it really happened over a short period of time. So this was all within a matter of a few months. And then this is an example of after we switched to sequencing around 2013. This is an example of an outbreak that was linked to deli sliced meats and cheeses. And what I want to point out on here is the date axis goes all the way back to 2016. So once we started using sequencing and having that greater degree of precision and ability to match isolates over time, we are able to then link outbreaks over a span of many years, especially for an organism like Listeria that can really be persistent in environmental and food processing settings. And what we found with the transition to sequencing for Listeria was that we were actually identifying and solving more outbreaks, which led to more recalls and pulling contaminated food off the market. But what we were finding is that we were having fewer cases per outbreak. So we were able to detect outbreaks quicker, solve them faster, and reduce the number of illnesses associated with Listeria. So after the successful pilot with Listeria, Salmonella was the next organism that went. So I want to talk about an outbreak that happened on the bubble when we were moving between PFGE and into sequencing, and we were actually doing the two technologies side by side. So we had 4 cases of Salmonella reported to the health department, all from the same county. 2 of them were reported in fall of 2017, and then 2 of them in May of 2018. And our typical cutoff for our PFGE investigations had been that cases had to occur within 60 days. And so using our normal protocols, we would have considered the 2 cases in May to be unrelated to the 2 cases in the fall of 2017. And we would have only investigated those 2 together. But because this was a really rare PFGE pattern, we had almost never seen it before, I had an inkling that maybe this really was not 2 separate clusters of illnesses, and maybe there was an ongoing linkage between these 2 clusters of 2. So at this point, we had the sequencing data, we just hadn't been using it. So I decided to go look at the sequencing data. And this is a phylogenetic tree pulled out of the NCBI's pathogen detection portal. It's a really great tool online where all of the public health sequencing data for enterics goes into this public NCBI repository, and you can pull up isolates and build trees with the isolates that you're interested in looking at. So when we looked at these 4 cases on the tree, we noticed that they all clustered together. And the other thing that we noticed with the isolate there with the star next to it, that was not one of the 4 isolates that we were considering part of the cluster. And when I went and looked that one up, it was also a Washington case, also from the same county as the other 4. And the reason that we hadn't picked it up before was because it had a slightly different PFGE pattern. So again, that was one of the limitations. PFGE, if your banding pattern was a little bit off, it would get called a different pattern name, and you wouldn't recognize that it really was closely related to other cases. And one really neat thing and helpful thing about that particular isolate that we found was that one had actually been associated with a solved outbreak the previous spring. So we were able to look at the information from that outbreak and say, hey, that one was associated with this rotisserie chicken outbreak at a chain store. Maybe we should focus on that area for these current cases. And so we did investigate these 4 cases together as one outbreak. And when we asked about that specific chain store that had been associated with the previous outbreak, everyone started saying yes, and actually said yes to shopping at the exact same location of the store. And we were able to zero in not on a particular food item, but everyone had some association with something from the deli. So environmental health did a lot of work with the store with checking their safety procedures and cleaning procedures in the deli. And we ended up having a total of 7 cases spread out almost over a year, indicating that there was likely some persistent environmental reservoir in the facility. And after taking all these mitigation steps, we flagged these isolates in the database, and I watched them like a hawk for a couple of years, and we never saw any additional ones pop up. So it was great that we were able to mitigate the outbreak and then watch over time to make sure that it really was taken care of. So, show of hands, who knows what kratom is? I've got one. I did not either. So we had a mystery cluster of illnesses in Washington state of Salmonella okidii, a weird serotype, and we could not figure out what they had in common. It was a really weird signal. Some of the people reported eating a lot of meals like at gas stations and buying stuff at on the go stores like that. And then another group in the outbreak cluster reported shopping at a lot of like health food stores and vitamin stores and stuff. And we could not figure out what do those 2 things have in common. And then I got a call from a colleague at the CDC that said, hey, can Washington hop on this conference call in a minute? We're talking about a kratom outbreak and we think you guys are involved. Said, a what outbreak? So I learned very quickly, kratom is it's a herbal supplement that comes from a plant generally grown in South Asia. People use it as a mood stabilizer, for pain, for many other things. And other states had been investigating an outbreak, and they had figured out that kratom was the likely source. They had gotten kratom into their public health lab, who then did testing for salmonella. And what they found was not just the salmonella that matched their cluster, they found a multitude of salmonella species in this kratom that they tested. And one of them happened to match our unsolved Salmonella okidii cluster. So once we had that clue, we were able to go back to our health food store eaters and our gas station store eaters and specifically ask about supplements and then specifically kratom. And sure enough, once we knew what questions to ask, everyone started reporting, oh yeah, I do eat that, or take that supplement. So that was a really cool way that we went from cluster to lab to that lab data, then solving multiple other unsolved clusters across the country. So this is another outbreak that happened in the PFGE era. So here's our DNA fingerprints. But this one started off with just a single case of salmonella reported, and the individual lived on a farm. And when the local epidemiologist contacted them to do the investigation, the person on the farm reported that they had had an abortion storm among their cattle around the same time, a little bit prior to their salmonella illness. And so we worked with the agriculture lab at the state, who actually had samples of the calf fetuses, and they did testing and were able to identify salmonella in the cattle fetuses. And then we were able to compare that to the human isolate, and it showed identical banding patterns by PFTE, indicating that they were likely due to a common source. And the interesting piece, after further investigation of how did this happen, was reporting that they had spread uncomposted, so just raw chicken litter, on their fields that the cattle were then grazing on. And we were able to get some samples of the leftover chicken litter and test that for salmonella, and sure enough, that came up positive. And you can see that the 3 poultry litter isolates, the 2 cattle fetus isolates, and the human ones all showed identical PFTE banding patterns. So this is kind of the holy grail of outbreak investigation. If you can get the clinical sample, an animal sample, an environmental sample all to tie together, it's a pretty cool way to really be sure that you're getting the story straight. So I don't want to just talk about bacteria, although I do tend to think they're a little more fun, but we'll talk about viruses too here. So one project that I worked on when I worked at the State Public Health Lab in Virginia was doing norovirus sequencing. So norovirus sequencing at that point, and I believe still now, was done just using Sanger sequencing of a small portion of the genome. So that's what I was doing in 2014. And most of the time we were using it just for surveillance purposes of what different genotypes of norovirus are going around. But there are instances where norovirus sequencing has actually been used to solve a foodborne outbreak. So norovirus is actually one of the most common causes of foodborne outbreaks. Most of the time, it's not because of the food itself, it's because food handlers, through improper hand washing, unfortunately, can transmit to the food as they prepare it. But in this instance, it was an outbreak associated with raw oysters. And so the health department received multiple complaints from different parties around the state where multiple people had gotten ill with norovirus after consuming raw oysters. And they were able to trace back using the tags associated with the shellfish and figure out that all these disparate outbreaks from around the state were tied back to one particular shellfish harvesting site. And when they did an on-site investigation, they went up and down the shoreline actually looking for any sources of contamination where norovirus may have gotten into the water where the shellfish were being grown. And what they found was one failing septic system that happened to be uphill from the growing site. And there was a drainage channel that provided a nice way for the water to flow down out of the failing septic system into the water where the shellfish were growing. And they were able to get, this was a colleague right before I came to Washington State, they were able to get samples of the septage out of the septic tank, out of the shellfish, the leftover shellfish, and then out of the people, and again compare all of those sequences of the virus and found that they were all identical. And so that really, again, wrapped up the story that we thought this was what was happening based on the epidemiologic evidence, but it was great to have the lab evidence come in and solidify our hypothesis. So as I mentioned, I was working on norovirus sequencing in 2014. Another thing that happened in 2014 was the Enterovirus D68 outbreak. And from mid-August to 2014 through January of 2015, there were at least 1,395 people confirmed with EVD68, but there was very little clinical testing that could actually identify it at that time. So we know that that was a severe undercount of how many cases were actually occurring. So Enterovirus D68 is one of, there are over 100 different non-polio enteroviruses. And so since I was working on norovirus sequencing, I asked my lab director if I could pivot to working on enterovirus sequencing to see if we would be able to, for our state, actually identify this virus in the state. And so the one advantage of having me work on that is that the CDC's protocol was very similar to what I was doing for norovirus sequencing, where it was a Sanger sequencing protocol, just of a small region of the virus. And so it used the same equipment that I was already using and we had in the lab up and running. The downsides of using this test were that it is lower throughput than if we had developed, say, a specific real-time PCR assay for EVD68, and it's a multi-day procedure. But the pros kind of outweighed that, in that if you just did the real-time PCR, you would just get a yes/no answer. And we know there's a lot of enteroviruses out there. It's not just EVD68 going around. So by doing a sequence-based assay, we are able to actually identify in those samples that weren't EVD68 what particular enterovirus it was. And one of them happened to be associated with a case of acute flaccid paralysis and this was my favorite enterovirus. I hope everyone has a favorite enterovirus. Mine is Enterovirus C105. So I think that was the most unusual one that I identified, and the University of Virginia clinical team that worked on this actually wrote it up as a case report on this patient. So that was really exciting to see our public health results feeding into clinical case management and knowledge dissemination. So we found a whole lot more viruses beyond just EVD68. So another outbreak that I worked on was, most people when they think of don't eat raw cookie dough, think first of salmonella and eggs. But this one was an outbreak linked to E. coli in flour. So there's multiple ways that you can get sick from cookie dough. I won't tell you whether or not I still eat cookie dough anyway. But this was a really large outbreak spanning multiple states. You can see it was all across the country. So we were all working really closely together across the country. CDC was coordinating, and they got to the hypothesis that it was caused by General Mills flour. And I was working in Virginia at the time, and we had one particular case that did not report any exposure to General Mills flour. We couldn't figure it out. How did this person get this outbreak strain? They don't match any of the food exposures. And this is an excerpt from the CDC website, and you'll see at the bottom there, 3 ill people, all children, reported eating or playing with raw dough at restaurants. So on one of our conference calls, one of my colleagues from another state said, hey, we figured out how one of our cases was exposed. They went to a Mexican restaurant. The Mexican restaurant would give out tortilla dough balls for kids to play with while they were waiting for their food. So we went back and called our case back because we had just asked about everything you had eaten. We didn't ask whether you played with dough balls at a Mexican restaurant. So once we were tipped off to that, we went back, and sure enough, the answer was yes. Sent environmental health out to collect the information for the flour, and it matched the recalled lot. So that was an example of how iterative these investigations are, especially the multi-state ones where we're all working together to try and solve what's going on across the country. All right, so I'm going to pivot to SARS-CoV-2 now. I didn't want to spend the whole time on this because I think we're probably all tired of it, but I think it's a really good story for public health and how it's being used in the current day. So there are many purposes of SARS-CoV-2 genomic surveillance in public health. It can be used to detect outbreaks, investigate outbreaks, especially in specific locations. For example, answer questions like, is a hospital outbreak a single introduction or multiple? Or are cases in schools being passed around at school, or are they just matching what's going around in the community, and it's coincident that they all happen to be in school too? And you can monitor transmission dynamics within and across communities, so between universities that are nearby or universities and communities. So there's a lot of ways that this data can be used to understand how diseases and how SARS-CoV-2 specifically is moving through the community. And then one thing that became really critical was monitoring for emerging variants. So the US initially lagged pretty far behind. So this is a chart of all of the available SARS-CoV-2 sequences in public repositories. So you can see through the entire first year of the pandemic, it almost doesn't even register. It was not a priority of public health, and not a whole lot of sequencing was being done. In April of 2021, there was a specific federal effort to ramp up sequencing, and you can see that since then, the number of sequences available for US cases has really skyrocketed. And it's been really exciting to see that federal investment and then states taking up the lead as well. So one of the kind of persistent issues, in addition to just the issue of not enough sequencing happening, trying to take that sequence data and then connect it with what's going on in public health is a piece that can be really challenging, especially with the US health system. And then even the US public health system is very fractured, where there's local health jurisdictions, there's states, there's 50 states. So it's trying to herd a lot of cats to get any sort of unified data set created. So one of my goals in Washington was to take in all the disparate sequencing data sources and bring them together to create one data set across Washington state that we could then use for different analyses. So trying to link the viral data with the epidemiologic and clinical data so we can make fuller use of it. And so this is a busy slide, but basically what we wanted to do was Washington Disease Reporting System is our epidemiologic surveillance database. So that's when a lab tests and gets a positive for SARS-CoV-2, they send a message electronically to the state health department, and that message lands into this WDRS system. So this is where all of the clinical and case data is held, and this is where, if you hear, reported case counts daily or things like that, this epidemiologic surveillance system is where that data lives and gets reported out from. So we wanted to take the viral sequencing information and actually link that into our surveillance system. And one of the challenges was there was many different labs. There was multiple academic labs that were doing sequencing. There were, CDC had contracted with commercial testing laboratories to do sequencing. Our public health lab was doing sequencing. So the biggest challenge was setting up the pipelines to get the data from all these over a dozen different labs, get it transformed into the right format, matched up to the right case in the surveillance system, and then it could finally be entered and be available for analysis. So I want to talk about a couple of the outputs of the work that we did on that. And one of them, a main one, is our SARS-CoV-2 sequencing and variants report. So this gets published on the Department of Health data dashboard for Washington State on a weekly basis. And it's generated in R Markdown. So because we're getting all of our sequence data into our one main database, we're then able to access that database programmatically, pull the data out, and create the report, so we can just push a button and run it weekly and have updated information. So that was really important for sustainability, that we couldn't be manually curating the data each week. So some of the things that we show on our report is the number of specimens sequenced by month, and then also the percent of COVID cases that represents. So you can see that some months, April through July of 2021, we were doing great, sequencing almost a quarter of the cases across the state. That was, of course, the lull when there were very few cases. So it made it a lot easier to get a high percentage. You can see in December, January of 2021 into 2022, during the Omicron spike, where there was just so many cases out there, Even though we were sequencing over 7,500 specimens a month, it was still not even a drop in the bucket of how many cases were occurring. So another thing we share in the report is looking at the percent of specimens sequenced by county. And this was really important because we wanted to make sure that we were doing as much as possible to get representation from around the state, because what's happening in Spokane or Whitman is not the same as Clallam or King County. So because testing can happen anywhere and there's so many different labs, it's really challenging to balance exactly getting the right number of specimens from every county. But we did really make concerted efforts to make sure that we were getting geographic coverage across the state. And this is an example of where we showed the proportion of the variants that we were seeing in the state. So you can see that currently it's all Omicron, but the BA2 subvariant of Omicron is really starting to increase just in the last 3 weeks. So that'll be something to watch over time to see as BA2 increases, does that have any effect on case counts or things like that? Another thing that gets shared in the report is the looking at known reinfections. So the definition for a reinfection for SARS-CoV-2 is someone who tests positive at least 90 days apart. So if you test positive and then at least 3 months later you get tested positive again, then you actually count again as a new case. And those will be called reinfections. So looking at this chart, September, October, November are pretty boring because all of the first column in the graph is the reinfection cases, the second column is the comparison, and that's our sentinel or just randomly selected specimens. So the first few months are pretty boring because everything everywhere was Delta. But then in December, as we saw Omicron emerge, you can see that among our sentinel cases, 58.9% of them were Omicron, but if we look at our reinfection cases, 88% were Omicron. So that gives an indication that there's likely more immune evasion in Omicron, where it's more easily able to reinfect people who had previous infections. And then again, looking at a similar type chart, this is for looking at the proportion of variants among vaccine breakthrough cases. So these are among individuals who had at least 2 documented doses of vaccine and then had a confirmed positive after, at least after 2 weeks past their last vaccine dose. And you can see that, again, it's all Delta for a while, but once you hit December, there is a little bit, it's not as dramatic as the reinfections, but we do have a higher proportion of the December vaccine breakthrough cases were attributed to Omicron infections versus the proportion we saw among our sentinel cases. And then this is another table. The table in the actual report is really busy, so I pulled out just a few of the data. So one thing that gets shown is the symptoms by variant, and most of them are fairly similar between Delta and Omicron, but I highlighted 2 that really were different, and that is for sore throat. 38% of people with Delta, when the health department called and asked about their symptoms, 38% of people infected with Delta reported having a sore throat, whereas 60% of people with Omicron. And then also with loss of taste or smell was 42% for Delta and then 16% for Omicron. So this goes along with a lot of the anecdotal evidence that I had seen in the news and stuff about more sore throats with Omicron. And so this was a way that we were able to share our population level data on a regular basis, which really lined up with some of the anecdotal data physicians and others were reporting. So another output, in addition to that weekly report, is through special analysis projects. So one thing that was a little bit unique in Washington and in the Northwest in general, actually, is the wide diversity of variants that we've seen here over time. So a lot of the best analysis and data has come out of the United Kingdom. where they have a really strong genomic surveillance program. But in the United Kingdom, they basically had Alpha, Delta, Omicron. Whereas in Washington State, we had Alpha, Beta, Delta, Epsilon, Gamma, Iota, and Omicron. So we wanted to, because we had that wide variety of variants, and we were able to link that back to our clinical and epidemiologic data, we worked on a project with the University of Washington and the Fred Hutch Cancer Research Center, as well as the Institute for Disease Modeling, to do an analysis of the association between SARS-CoV-2 variants and risk of COVID-19 hospitalizations. So this analysis is currently posted as a preprint on medRxiv. But what we saw is that line there with the 1 represents the hospitalization risk for people with ancestral COVID lineages. So pre-alpha, that was the hospitalization risk. And then you can see that 4 of the variants had significantly higher risk of hospitalization. And this was after adjusting for sex, age, calendar week, and vaccination status. So alpha was between about 1.6 times higher risk of hospitalization, and then gamma had the highest estimate at 3.2. And then you'll see Omicron down at the bottom there, which the point estimate is just below 1, but it's not significantly different, with a pretty wide confidence interval there. So this was a really great collaboration. The writing was led by a PhD student at the University of Washington. So really great to combine the analytical and research capabilities of the university with the data sets that public health was able to gather, and then we were able to combine to work on this project. So as Clint mentioned, I'm not going to talk a whole lot about this because I started my new position in January of this year, working at Biobot Analytics, focusing on wastewater surveillance and sequencing. But I did want to mention just a little bit. So this is some data off the Biobot website, showing in the dark blue line, the heavier line, is the average wastewater levels of SARS-CoV-2 for all their testing sites. And then the bars and the lighter blue is the average of case counts. So you can see that in general, the wastewater levels track very well with reported case counts. And what's kind of interesting, if you look at the Omicron peak there, you can see that there's a greater divergence between the height of the reported cases and the height of the wastewater cases. And that likely is due to, as Omicron hit, the availability of testing became limited, the availability of at-home testing became more common. So it's likely that the proportion of true cases actually being reported and showing up in the public health case counts decreased during the time of Omicron, but the wastewater would still capture those unreported cases. And then another really neat use of the wastewater data is being able to estimate the variant fractions using the wastewater data. So that's a new and upcoming and really exciting area. It can be very expensive to get enough clinical sequencing to try and get a good bead on what proportion of variants are out in the community. But by doing the wastewater, you can get that estimate without having to sample hundreds of people. So some conclusions. Advanced molecular detection techniques really play a critical role in moving infectious disease surveillance and investigations forward. And one of my key takeaways from the past 2 years working on COVID especially is that close cooperation between disciplines is really necessary to make the best use of the information. And then also the public health workforce needs to increase capacity for molecular surveillance. And this needs to happen through training existing staff and then also recruiting new staff with cross-cutting capabilities. So this is bioinformaticians that have interest in epidemiology, and data scientists that have interest in biology, and molecular epidemiologists that have interest in both the molecular biology and the epidemiologic investigations. So there's a lot of room for growth and a lot of room for interdisciplinary collaboration and really cross-training people to have them maybe have a specific focus area, but also learn the lingo of a different field so you can collaborate really well across the spectrum. And then Clint, I have a few more appendix slides I can do, or we can do questions now.
You've got time.
All right, I will keep going then. So I wanted to walk through a few tools that I found really helpful for visualizing genomic and epidemiologic data together. One of them is called NextStrain. So this is comes out of a few labs around the world, one of them being Trevor Bedford's lab at Fred Hutch. And NextStrain is a really great tool that allows you to build these really beautiful and interactive phylogenetic trees. The CDC actually maintains NextStrain builds for all 50 states. So this particular one is actually a Idaho-focused build that if you go to the NextStrain link there at any time, it'll be updated with the most recent sequences from Idaho. So these just get pulled out of the GISAID database. So if any Idaho institutions upload their sequence data, it'll get pulled in for visualization. And then some states maintain their own NextStrain builds. This one's really messy because it's all Omicron, and it's from Washington State, who has done a lot of sequencing. So some states maintain their own public-facing NextStrain builds as well. And one of the really great features of NextStrain is that you can actually drag and drop your own metadata onto the public-facing NextStrain trees. So in this example, I just went to a public NextStrain tree and took a CSV file, or TSV, that had a list of this specimen ID went with outbreak A, and this one went with outbreak B. You can drag and drop that onto your browser, and it doesn't send it back to NextStrain. It all works locally within your browser. And then you can do things like label or color your tips on the tree based on that additional metadata that you dropped on. So that's been really great for public health, because I could send a local health department a link to our public NextStrain tree and attach a Excel file and say, hey, drag and drop this Excel file on, and then you'll be able to see your outbreak live and interact with it. Another tool that leverages NextStrain trees is called Usher. This is made out of the University of California, Santa Cruz. And what they do is they have a gigantic phylogenetic tree of all 4 million plus SARS-CoV-2 sequences that they refresh nightly. And then you can either upload your own sequence files, or you can just enter the GISAID IDs of your specific sequences that you're interested in, and then it will show you a NextStrain tree with your sequences of interest placed on that tree. And then it'll show you a gigantic tree, so you can see kind of where they fall in the big picture of things, and then you can look at smaller subtrees and really drill in and answer questions like, I have this particular isolate from my jurisdiction. I want to see what the most closely related isolates are. So you might look at your particular one and go, oh, wow, this clusters with all genomes from the UK. Maybe this person traveled, or they were associated with someone who traveled. So you can start to really drill into your data and make connections. And then another tool that I really like is called Microbetrace. And this is put out by the CDC. And again, it just runs in your browser, so you can use more sensitive data and it doesn't get sent out to process. It all stays locally. And one of the neat things about Microbetrace is that it has this network view. So each of the pluses and circles here represents a SARS-CoV-2 case. And you probably wouldn't make it this busy in real life, but I was trying to show all the different options. So if you look just at the shading under the polygon heading here, this is a made up classroom outbreak, but this shading around here represents that these 4 cases were all in classroom 1. These 3 cases were all in classroom 2. If we look at the links, we have these ones all had genomic links. So you can set your threshold of whether your genomic link is, they have to be identical sequences, or how many differences you allow to draw the genomic links. And then one of the neat features of this is that you can actually add linkages based on other information. So we can see that this case A1 and case A2 that are in separate classrooms are linked as household members. So you can represent both the epidemiologic linkages and the genomic linkages on the same figure. And then you can color, so green would represent our probable cases. So those would be people who either had an antigen test or were just symptomatic but not tested. Those would be considered probable cases, and you can see that those ones are not genomically linked to the other cases because they wouldn't have been sequenced. And then you can use the shape of the nodes as well to denote other data. I have these shaped by sex right now. So you can really display a lot of dimensions of data at the same time. And I found that in presenting data using phylogenetic trees and the network views, some people really have an easier time latching on to the network views and what that means. And we'll end on, this is a poster from a conference a couple years ago that unfortunately got canceled for COVID. So I'm going to show it here because it didn't get to get shown in March of 2020. Again, featuring Microbetrace and all of the different microbetraces, not just the network view. You can do phylogenetic trees, you can do 3D networks, bubble diagrams, maps, matrices, flow diagrams, and other things. So really finding unique tools to be able to communicate the complex sequencing data in a visual way can be really helpful. And that is the end of my talk. I love to talk to people, so you're welcome to reach out to me. That's my email. If you forget anything else and remember my first name, since it's a weird one, I managed to get Krisandra.com at one point and it just redirects to my LinkedIn. So if you forget anything else and want to find me, you can find me that way. Thank you.
We've got a few minutes for questions.
Yeah.
So I'm going to see if tomorrow I'll keep the base for the question. I was curious, early on you said that there were some cases that were Salmonella or Listeria that were linked. They had the same PFGE pattern, but when you cluster them based on genome sequencing, they were not.
Yeah.
So can you elaborate a little bit on that? How they explain that, or the other way around actually? Is that they have different PFG patterns that they clustered based on sequencing?
Yeah, and we actually saw it both ways. So we saw Salmonella Enterovirus was the worst on PFGE because we would just see, gigantic PFGE clusters of the same pattern of Salmonella Enterovirus. But then if you sequenced them, they really broke out into a whole lot of different clusters. And then we saw it the opposite way of where you would have different PFGE patterns, but then when you sequence them, they would all cluster together.
So that's the one I don't understand. Do you have any deeper explanation for that? Yeah. Why would that be? They have different PFGE patterns?
I think PFGE was not a perfect technology and just had some limitations, and we've seen that the sequencing data tends to line up better with the epidemiologic data. So I don't know the underlying biological reason why some of the PFGE, and some of it is just also the resolution like the way that patterns would get called on PFG was literally uploading a picture to CDC, and then they would draw the lines in. And if your band was slightly shifted, then it would get named a different PFGE pattern. So it was involved some interpretation as well. So they do whole genome sequencing. The analysis methods vary. Like CDC often uses core genome MLST for doing the foodborne investigations, and then FDA usually uses SNP analysis. So it kind of depends on the agency. Yeah.
Great talk, I loved it.
Thank you.
I'm wondering if you see any correlation between the appearance of variants in SARS-CoV-2 that correlates with public health policy. So, for example, I don't know, in Washington, maybe they're not changing their public health policy, but I guess that's the question. Public health policy, have you seen any correlation between variants?
Yeah, I don't think I've seen a lot of correlation between the appearance of variants in public health policy. I think policy tends to lag the variant's appearance quite a bit. So the new variants will tend to emerge and start spreading, and by the time any policy implementation is made, it's kind of the horse is out of the barn to a degree at that point. Yeah.
I have a question. To what degree is PFG still used? Is that still like an initial frontline thing that's always done, and then sequencing is done sometimes secondarily, or has it actually changed where PFG is not done in the lab in that instance?
Yeah, great question. So the question was about whether PFGE was still done, and I don't know of any PFGE still being done. It might be in some weird organisms, but for example, all of the enterics like Salmonella, E. coli now is all done by sequencing, Listeria, and even the current Chronobacter and Salmonella outbreak in infant formula is being done through sequencing as well. So it's really kind of replaced PFG completely. Yeah.
Yeah, I have a question, and so this follow-up on the question. I was wondering with biology and sequencing, because the strengths may be very different or very similar in different approach, whether it's like SNP or genome, at which point, like it can be called an outbreak, or at which point you draw the line where, say, those strengths are totally the same outbreak, it doesn't have to be identical, or if there's room for a little bit of variation.
Yeah, that's a great question on and something that we wrestle with a lot. So for the foodborne pathogens, as I mentioned, CDC for cluster calling generally uses core genome MLST, and what they did was look at they did in parallel for many years of the PFGE and the sequencing, and then they were able to look at all of the confirmed PFGE outbreaks and then look at the thresholds for the core genome MLST to line up where those cutoffs are. And I don't have them off the top of my head, but it's not zero. So there definitely is some wiggle room, and that is something that is constantly refined over time. And that's something that we really struggled with on the SARS-CoV-2 interpretation of, this is brand new virus, we're brand new at sequencing this. How close is close enough to say that some two cases are likely epidemiologically linked? And I don't think that one's really been solved yet. Yeah.
You're showing the proportion of variants over time in Washington. It's right here. Is that normal for the variants to rise?
No, I don't think so. I think that's one thing that has been really unique about COVID, like talking to people that have studied flu for a long time. They tell me that it takes, usually a couple years for a new flu strain to emerge and sweep. So to see something like Omicron that emerged and swept to almost 100% within a matter of really 3 to 4 weeks is really striking and pretty amazing.
Come back and look at like historical data, or is there tables from outbreaks as well, kind of monitoring them?
Yeah, I am not aware of freezer diving to really, I'm sure there are projects out there that have tried to reconstruct historical data like that. Yeah.
Are you mentioned the changes in like symptom reporting between stuff that is on
Yeah.
but that was within just Washington State.
Correct. Yeah.
Are those results consistent with other states, either nationally or globally?
That is a great question, and it's hard to answer because most states either don't merge the data to be able to look at that or don't report it publicly. So I'm only aware of New Mexico also has a report that breaks it down by variant. That's actually where I took the idea from. And I haven't checked theirs recently to see if they were seeing the same pattern. Yeah.
So with you showed us a figure on the hospitalization comparing to the original strain of variant and all the others. So you corrected for like vaccination and other demographic things. Do you happen to look at like the differences of that with just vaccinated individuals and not vaccinated individuals, the difference among hospitalizations?
We didn't look at it stratified that way. I think because we ended up with such small numbers in some of the groups when we stratified it.
Any other questions? Okay, great, thank you.